Hi everyone, my name is Nina Derschlag, and for today's interactive presentation, we will be going over how heme is bound and released. An essential component here is oxygen, hence it being vital to our survival, along with it being the terminal electron acceptor in which is vital for the Krebs cycle. And so when we breathe in air, we get oxygen and many other gases. However, let's focus on oxygen. It's going to enter the lungs, and from the lungs, it's going to get picked up by the red blood cells, which will then transport it to the rest of the body. So here in this figure is the oxygen transport cycle. So number one here, we got the lungs, and it's got the oxygen from the pulmonary alveoli that are going to bind to the heme. And then we got the step 2 erythrocytes. The red blood cells are going to carry the oxygens from the lungs to the rest of the cells, where then in step 3, the oxygen is going to release them to the tissue cells. So this is all done through diffusion. Within the lung, oxygen is going to diffuse from the alveoli into the pulmonary capillaries, driven by the gradient between the partial pressure of oxygen in the alveolar space, and that in the deoxygenated pulmonary capillary blood, a protein called heme in the red blood cells then carries the oxygen around our body, and so oxygen must be transported in the blood from the lungs because of this partial pressure of oxygen, which is relatively high. In TOR, it's about 100. Then the tissues, where the partial pressure of oxygen is going to be much lower and typically be around 20 TORs. Red blood cells. So as far as their shape, I have an image that I attached here. They are a biconcave disc, and they're erythrocytes. So erythro is red, cyte is cell, and they are what are going to help transport the oxygen. So a red blood cell is interesting cell. Unlike other cells in our body, it's got lipid bilayer, then it has a cytoplasm. However, what it doesn't have is a mitochondria and a nucleus. And so when red blood cells are made in the bone marrow, they do have a nucleus that would be the pre-red blood cell, since the nucleus has a DNA and DNA codes for proteins, and proteins will then carry out those functions of that growing cell that's going to need to grow. So a red blood cell is then going to mature. It has to lose its nucleus that it'll allow it to have more space to then carry a very critical protein known as hemoglobin. So as far as red blood cells, we know that there's no nucleus, we know that there's no mitochondria, in which is the powerhouse of the cell and is where the bulk of ATP in the cell is being made, but the mitochondria, which has the electron transport chain, required oxygen. So we may ask ourselves, do we really want the cell that carries oxygen to other cells to potentially eat up oxygen along the way? Definitely not. And so red blood cells rely on glycolysis for their energy to focus on carrying oxygen to other cells. And if the cell isn't taken up by a nucleus and isn't taken up by mitochondria, it has so much more room for the protein hemoglobin, which is what we essentially care about. And 35% of red blood cells is what's occupied by hemoglobin. Hemoglobin is also the protein that physically carries the oxygen. Hemoglobin, what is it? So according to this figure here, hemoglobin is a protein in red blood cells that binds to oxygen in the lungs and carries the oxygen to the tissues. It's known as a tetrameric protein, which consists of 4 subunits, each having 1 polypeptide chain and 1 heme group, 2 alpha subunits, and 2 beta subunits. Oxygen binding induces substantial structural changes at the iron sites in hemoglobin. So the binding of oxygen molecule at the 6 coordination site of the iron is substantially rearranges the electrons within the iron so that the ion becomes effectively smaller, allowing it to move into the plane of porphyrin. And so this change is taking place in electronic structure and magnetic properties of hemoglobin, which are the basics for functional magnetic resonance imaging. On oxygen binding, there are substantial changes in quaternary structure that correspond to the T to R state transition. In lungs, oxygen binds to heme, and in capillaries, oxygen dissociates from heme and mixes in blood. A decrease in pH shifts the standard curve toward the right, and so this occurs because at greater hydrogen ion concentrations, various amino acid residues such as histidine stabilize deoxyhemoglobin in the T state. The T state has lower affinity for oxygen than the R state, so with increased acidity, the hemoglobin binds less O2 for a given PO2 and more hydrogen, which is known as the Bohr effect. And so as CO2 is acidic in nature, when it interacts with water, the pH of capillaries are then decreased. How heme is bound. So the iron atom of the heme group is attached to the nitrogen atom of the histidine amino acid of the protein. The 4 globular proteins consist of 2 alpha polypeptide chains and 2 beta polypeptide chains. They are attached with the help of hydrogen bond, ionic bond, and hydrophobic bond. How heme is released. So upon the hemoglobin that's released from the red blood cells to the circulation, it then gives a rise in production of different hemoglobin redox states. And heme, which can act as damage-associated molecular patterns, is best characterized in which it targets different immune and non-immune cells. How CO2 interacting with water can affect the pH of the capillaries. Here, carbon dioxide is influential in regulating pH in water. The amount of CO2 in a solution is one way to determine the pH. The more CO2 in water, the lower the pH. So carbon dioxide influences pH of blood by reacting with water to form carbonic acid, which can dissociate to form a hydrogen ion and a hydrogen carbonate. Increasing the concentration of CO2 in the blood therefore results in more hydrogen ions, thus a lower pH.